Hi kiddos. Welcome to your little video lecture today. It's on DNA, which we could also call the blueprint of life. If you look at the picture of the dog, you can see some very distinct characteristics that are being expressed. When we say something is expressed, that just means that the DNA is coding for proteins which then become a visible trait. For instance, you can tell that this dog has curly hair. There are some traits that are expressed that you can't see, like whether or not you're a PTC taster. PTC stands for phenylthiocarbamide, and what this means, it's just a chemical, and some people have receptors in their tongue where they can taste it and some don't. If you're curious about whether or not you're a PTC taster, check with your teacher. I bet you she has some PTC paper for you. The DNA codes for all of these things, right? It stands for deoxyribonucleic acid, and in eukaryotic cells, it's found packaged into chromosomes and inside the nucleus. Segments of DNA code for specific traits, and we call those chunks of DNA that code for traits genes. Once we determined that DNA was the molecule of inheritance, it was really important to understand its structure. Determining the structure of DNA is attributed to James Watson and Francis Crick, oftentimes called Watson and Crick. However, they did not do this alone. Oftentimes people will discuss the unsung story of Rosalind Franklin, who did X-ray diffractionation images of DNA and helped them determine the structure. But it wasn't just her. In addition, Chargaff was a Russian scientist, and he determined that if you had 20% adenine, you'd have 20% thymine. If you had 30% guanine, you'd have 30% cytosine. So what he figured out was that the guanine and cytosine must be pairing up in some way. So now we know that adenine always pairs with thymine. So if there's 20% adenine, there has to be 20% thymine. These observations became Chargaff's rule, where it stated that adenine will pair with thymine and guanine will pair with cytosine. I like to think of it as the straight letters will pair with the straight letter and the curvy letter pairs with the curvy letters. So if you had 15% adenine and 15% thymine, then you would have to have, that would equal 30. That means that you have 70% of G and C together. So 30% is A and T, then 70% would have to be G and C, and then you can take that number and divide it by 2 to make a prediction of how much guanine and cytosine you have. So Watson and Crick now knew that adenine and thymine paired and guanine and cytosine paired, but they didn't still know the structure of the DNA molecule until they saw Rosalind Franklin's picture. This famous picture right here showed them that it looked like there was rungs of 2 ladders, but as you went down the depth of the molecule, the ladders seemed to twist. That helped them come up with the shape of the double helix. A double helix is like if you took a ladder and then you just twisted it. And it looks something like this. This is a clearer picture for you as well. You can see here that adenine is pairing with thymine and guanine is pairing with cytosine. And what that does is because these molecules are large and this molecule is small, this molecule is small, this molecule is large, it keeps the rungs of the ladder even, so the twisting is also even. Remember, the nucleic acids are made out of nucleotides. Nucleotides are a 5-carbon sugar, a base of some sort, a nitrogen base, and a phosphate group. You should remember that from biochemistry. Here is an outline of the 3 parts: phosphate, sugar, base. When we drew pictures of them, I said you draw a picture of a house and you put a flag on it, and then this is your little puff of smoke coming out of the chimney. Hope you remember that. The sides of the molecule are phosphate and sugars that are bonded together with covalent bonds. But the 2 separate rungs of the ladder are held in between these pairs with hydrogen bonds. Now, if you remember from biochemistry, hydrogen bonds are pretty weak. We said it was like trying to hold me in place by pulling a piece of my hair, if you remember. So one piece of hair isn't very strong, but when you have lots of hydrogen bonds working together, they can make a difference. In this case, hydrogen bonds allow the DNA to unzip and rezip. All right, so what I want you guys to know is this right here is a covalent bond that stacks the sugar phosphate rungs of the ladder together. But I also want you to note that this side is running what we call anti-parallel. So this side is upside down to this side. So the two sides aren't exactly alike, but they're anti-parallel to each other. So you can see here the code is AGTACG. Here it's CGTACT. So they are not exactly alike, the two sides of the ladder, but they complement each other. You can see adenine is pairing with thymine, as per, and the hydrogen bonds are holding those together. I also would like you to note that adenine and thymine have 2 hydrogen bonds, while guanine and cytosine have 3 hydrogen bonds. Because we know Chargaff's base pair rules, if we have one strand of DNA, we can guess what's on the other side. So let's see if we can do this together. A would pair with T. T would pair with A. A would pair with T. T would pair with A. C pairs with G. A, T, T, A. G pairs with C. C pairs with G. And here, this one would be C, C, C. Think of the bases of DNA like letters, and letters form words, and then words form sentences. So when we say that DNA codes for instructions, it's coding in a different language that has only 4 letters. But your body can read that and make all of the proteins in your body. DNA is replicated in a semi-conservative manner, meaning that from the parent strand, each new strand separates. Remember I said they can unzip? Well, the hydrogen bonds that are there are pretty weak, and so it can easily be pulled apart, and then they just add new nucleotides. So like if this was A, T, C, G, G, C, then this would have the complement on this side, which actually would look exactly like this original parent strand. We call this type of replication semi-conservative replication. As we go through the next few units, you're going to hear about a different type of nucleic acid. It's called RNA. What I'd like you to know right now is that DNA, deoxyribonucleic acid, is a double helix. It has a deoxyribose sugar in its backbone. We call this the backbone or the rung of the ladder, and it's found in the nucleus. RNA is a single strand. It has 3 major forms, mRNA, tRNA, and rRNA. We'll learn about those later. But it has a ribose sugar in its backbone or along the rung of its ladder. And instead of bonding to thymine, A binding to thymine, it binds to uracil in RNA. So uracil is like RNA's version of thymine. All right, I'd like you to pause the video a little bit, and I would like you to please see if you can answer these questions. DNA stands for hopefully you've paused it. It stands for deoxyribonucleic acid. The shape of DNA comes in a double helix. Watson and Crick determined the structure of DNA. Adenine always pairs with thymine in DNA. The sides of a DNA ladder are deoxyribose and a phosphate group. Once again, why don't you pause and see if you can answer the following questions on your own. All right, the answers are guanine always pairs with cytosine. The complementary sequence to this would be G pairs with C, T, A, T, C. The 2 sides of DNA are held together by hydrogen bonds. DNA is composed of repeating subunits called nucleotides, and the 4 bases that make up the rungs of the DNA ladder are cytosine, adenine, thymine, and guanine. I hope you got them all correct, kiddos. Kiddos, you stuck with me. Thank you so much. I hope you're having a great time.